Good afternoon, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is CRISPR Cas9 Screening in Human Pluripotent Stem Cells for Target Discovery, and our sponsor is Twist Bioscience. Our panelists today are Dr. Robert Eyre, postdoctoral scholar of neuroscience at the Novartis Institutes for Biomedical Research, and Charles Joseph, product manager at Twist. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. Dr. Eyre, please go ahead.
All right. First of all, I'd like to thank GenomeWeb and Twist for the opportunity to present today. I'm a postdoc in the neuroscience department at Novartis, and the big goal for my project is to conduct genetic screens in human neurons. This would identify novel regulators of neurological disease and identify better drug targets for neuroscience. To accomplish this, I've been working to combine two emerging technologies. That's the CRISPR Cas9 system and Human Pluripotent Stem Cells. Human Pluripotent Stem Cells, or hPSCs, are incredible cells. They have the ability to self-renew and culture indefinitely, and importantly, they're pluripotent. And this means that they can differentiate into all three germ layers in a wide variety of more mature cell types. Importantly, these cells can be used to quickly generate neurons. All right. Now, despite these advantages, there's some clear challenges to working with these cells, and they're not commonly used for high-throughput functional genomic analysis. These cells are sensitive to a wide variety of stressors. They spontaneously differentiate and die. They require expensive media and daily feeds. Lastly, genetic manipulation techniques that are robust in other cell types don't work as well here. Given how well Cas9 has been performing in a wide variety of organisms, we thought we could address some of these issues and develop a platform that will allow us to conduct high-throughput genetic experiments. All right, so I'll have three sections of the talk today. The first section of the talk will be talking about CRISPR Cas9-induced toxicity in hPSCs. Okay, so we wanted to, because hPSCs are so sensitive, we wanted to develop an inducible Cas9 system, and this would give us control over when Cas9 could be active. The first system we developed was this shield inducible system. So it works by fusing a destabilizing domain to Cas9, and this targets it to the proteasome for degradation. So the protein is always being degraded in the absence of this ligand shield. When you add this small molecule shield, it stabilizes Cas9 protein, and you actually get an increase of Cas9. You can see this in the flag staining below here. Cas9 levels increase when you add shield. All right, so we next wanted to test how well does this system work. So we first started using synthetic CRISPR RNAs and trace RNA pairs, shown here on the left. These can be ordered from IDT, and they're just synthesized. We used lipids to deliver them to the cells. And the first gene we started knocking out was EpCAM. It's a cell surface marker in stem cells, and there's a robust antibody to stain for it. So on the right, you can see the mock-treated cells have robust staining of both EpCAM and OCT4, whereas the cells treated with the EpCAM CRISPR RNA lose the expression of EpCAM but are still pluripotent and have OCT4 expression. So the system's working. Now, there's a few minor problems with this system. First, the synthetic CRISPR RNAs are kind of variable. We're getting an average editing efficiency with one treatment around 50%, but it can be as high as 80%. And as low as 20. So it's variable, and it's probably because it's a transient treatment with the RNAs. And the other problem is that the shield system has kind of some leaky activity. So in the absence of shield, Cas9 is still active. So we wanted a tighter control over this system and a higher average knockout, so we switched to this DOX-inducible system. So we generated this DOX-inducible construct that we knocked into the safe harbor locus of our cells. We used the third-generation TET activator system to induce Cas9 expression. We also added an insulator to make sure that Cas9 wouldn't be expressed in the absence of DOX. To deliver the guide RNAs, we use lentiviruses, and these integrate into the genome of the infected cells, and we have a U6 promoter that will constitutively drive the expression of the guide RNA. Importantly, there's no Cas9 expression or activity when there's no docs, and that's shown here. So if you look at the mRNA levels or the protein levels, you can see there's no Cas9 in the absence of docs, and that's important for some of the studies we'll be doing later on. So we wanted to test how efficient this system was, so we designed 47 guide RNAs targeting 16 genes. We packaged the lentiviruses and infected cells in a 96-well plate. We grew the cells for 8 days, and then at that point we isolated DNA and used next-generation sequencing to quantify indels. And that's shown here. So in the control reads are marked by the white bars, and the in-frame and frame shift mutations are in blue. For the control cells that weren't infected, you see there's no mutations, whereas infected cells treated with docs, there's an abundance of mutations. Now the average for the 47 guide RNAs was over 90%. And if you look at the best guide RNA per gene, we have an 81% frame shift average. So this is quite remarkable efficiencies, especially for hPSCs, and we're really happy about this. But there was a problem. Now, at the end of the experiment, you could see that the density of the cells was all over the place. The non-targeting controls were completely confluent, but the cells that had been edited were sparse and variable. This is a big problem, especially for automated neuronal differentiation, which is density dependent. So for a large portion of the talk, I will go over and investigate what's going on with this apparent toxicity to Cas9 editing. Okay, so to get into this more carefully, we'll be comparing a non-targeting guide RNA that doesn't make a double-strand break to a targeting guide RNA that has a site in the genome to bind and creates a double-strand break. For many of the examples shown here, I'll be using a guide targeting MAPT, which is a neuronal gene that's not expressed in human pluripotent stem cells, and it's not required for fitness. So it's essentially a double-strand break. Now, if we look at these cells grown in culture with Cas9, you can see that cells, this is confluence on the y-axis, and then on the x-axis is days after dox induction. You can see that cells grown with Cas9 in a non-targeting guide RNA actually continue to proliferate quite nicely, whereas cells that are being actively edited with a targeting guide RNA decrease confluency over time. See that these cells are still stem cells, they're just far less of them, and they're kind of peeling off the plate. Now this is a representative image of what the cells look like after we change the media and remove the debris. Eventually, as we kind of get out to 10 days, the cells start to become resistant to the reagent because all of the sites are mutated and they start to grow normally and proliferate. So we can still use these cells, but there's a very acute toxic response during editing. Okay, so we're a little unsure about this, like this toxicity thing, you know, is it this particular reagent, or there's some off targets? So we wanted to really scale this up and see if this would hold true. So we decided to do a pooled CRISPR screen where we had a pooled lentiviral library with 13,000 guide RNAs all mixed together. It's targeting 2,600 genes. Now this was a large-scale experiment. We infected 200 million cells at 0.5 MOI. This would be big enough to be a genome-scale experiment in cancer cell lines, but because we had this toxicity issue, we decided to do a more focused library and have control conditions to really see what was going on. So we have 4 control conditions. The first 2 conditions right here are in the absence of Cas9. So we have the parental H1s, we have the dox-inducible Cas9 without dox. So these 2 cells are just infected with the guides, but they don't have Cas9 being induced. The last 2 conditions in purple and green are the dox-inducible Cas9 with dox or the shield-inducible Cas9 with shield. Now we grew these cells for 12 days, and every 4 days we passaged the cells and collected DNA for analysis. It became very apparent at the day 4 passage right here that there was a synchronous and systemic toxic response to Cas9 activity. You can see the 2 conditions without Cas9, the cells were growing normally, but the ones with Cas9 had far fewer cells. So for 13,000 guide RNAs, we saw the same effect that we saw with the MAPT guide RNA. So Cas9 looks to be very toxic to stem cells. All right, so next we wanted to look at the representation of the individual guide RNAs during this experiment. So as you know, these lentiviruses integrate in the genome, and what we can do is actually use PCR to amplify this region containing the spacer right here, and then we can basically use this as a barcode to quantify the number of cells with a particular reagent, and that's kind of a proxy for mutant cells. We infected the population with 1,000 guide RNAs, 1,000 cells per guide RNA, and we did this at a low MOI, so only one guide RNA was infected in each cell. And then we'll use NGS to detect changes in guide RNA representation over time. So here's a brief overview of how this works. So we do the PCR and the sequencing, then we normalize the counts, we aggregate the replicates, and then we calculate a log2 fold change. And for this example, we'll be basically taking our individual samples and normalizing it to the original lentiviral pool to calculate the log2 fold change. And that's shown here. So this is for the 13,000 guide RNAs in the library. We have a log fold change on the y-axis, and then we have the different conditions over time on the x-axis. And if we look at the two conditions without Cas9 in gray and black, you can see that there's really no change in the representation of the guide RNAs in the library over time. It remains constant, plus or minus 1 log2 fold change. If we look at the conditions with Cas9, you can see that there's a time-dependent increase or decrease in the representation of guide RNAs. And this is specific to the Cas9 conditions. So genes are increasing or dropping out of the population. So what's important is if you look at this day 4 time point, which is after the toxic response has already started, you can see that representation hasn't changed at all. So this toxic response isn't because of the loss of essential genes. It's more related to Cas9 activity. And we really confirmed this when we looked at the non-targeting guide RNAs in the screen. So these guides don't make a double-strand break, and you can see that they have an immediate impact on fitness, and they outcompete the cells with targeting guide RNAs that have double-strand breaks. So this really confirmed on a large scale that there is a toxic response to Cas9 activity in pluripotent stem cells. Okay, is this specific to hPSCs? We next took CRISPR screening data from 14 additional cancer lines and looked at the non-targeting guide RNAs, and we can see that there's a strong enrichment of non-targeting guide RNAs in the stem cells, but not the cancer lines. And this is fitting given that DNA damage response and apoptotic pathways are often mutated in cancer, so they won't have as severe of a response as human pluripotent stem cells. Okay, we wanted to get into the mechanism of this a little bit more, so we decided to do an RNA-seq. We'll be comparing a targeting guide RNA to a non-targeting guide RNA 2 days after double-strand break induction. We did a RNA-seq and differential expression analysis. And here's the result. So this is a volcano plot with a p-value on the y-axis and a log2 fold change on the x-axis. And you can see that a double-strand break triggered by Cas9 induces a lot of response genes. The number 1 most differentially expressed gene was p21, and this is no surprise because it's a known cell cycle regulator and it's involved in the DNA damage response. We also identified a number components of the intrinsic and extrinsic cell death pathways, which is consistent with the toxic phenotypic response. To really get at what was driving these transcriptional changes, we used a causal reasoning algorithm which generates and ranks hypotheses explaining the changes in transcription. And one of the top ranking hypotheses was P53. P53 had direct connections to 33 out of the top 100 differentially expressed genes. So it became very obvious that P53 was involved. Now this isn't a big surprise considering P53's well-established roles in the DNA damage response pathway, but at the time this was really important for us and allowed us to focus on just one thing. There weren't any reports of this in the literature. It could have been a number of things. We're expressing this 2-component foreign bacterial system in highly sensitive cells. So we really weren't sure what it is. So it was actually nice to find out that it was primarily because of the double-strand break response. Okay, if we look back at the non-targeting guide RNA enrichment and we look at the cancer lines, p53 is one of the most commonly mutated genes. About 50% of cancers have mutations, and you can see outlined in red we have the cancer cell lines with p53 mutations, and you can see that they tend to have very little enrichment of non-targeting guide RNA, so they have a very minimal double-strand break response. So this further implicates p53 in this toxic response. We really wanted to do a better job of this and do this in an isogenic line. So I knocked out p53 in our Inducible CAS9 line, and then I want to see if it was functionally involved, and that's shown here. So we're measuring confluence over time in this graph, and all cells are expressing a targeting guide RNA, and they're grown plus or minus DOX. So you can see that the control and mutant cells grown in the absence of DOX kind of grow at the same rate, but when you add DOX, the control cells dramatically die, whereas the mutant cells actually continue to proliferate despite having a double-strand break. And this really confirmed that p53 is responsible for the toxic response to Cas9 activity in stem cells. Now, if you consider all the things I've shown you, it looks like hPSCs would really rather die following DNA damage than actually repair or allow for a mutation. And we hypothesized that this was a major barrier to precise genome engineering efforts because the way that HDR is homology-dependent repair is commonly enhanced is by using Cas9 to create a double-strand break, and that allows your template to recombine and knock in a transgene or a specific mutation that you want to make. And it's well known that this does not work very well in hPSCs, so we thought that toxicity was really prohibitive in the cause of the low efficiency, so we wanted to test that. That's shown here. So we came up with this HDR assay. This is the OCT4 locus. We designed a dual nickase to make a double-strand break right at the stop codon of OCT4. And then we have this gene trapping plasmid, which has homology to OCT4. And what it does, it directly fuses this TD tomato to the OCT4 open reading frame, and it also drives the puromycin resistance gene. Importantly, all cells that are puromycin resistant or have nuclear TD tomato were properly targeted, so it's really easy to screen for proper targeting events. Now, we did this by electroporating the cells using expression plasmids for Cas9, the guide RNAs, and then we did this in the presence or absence of a plasmid that could inhibit p53. So this plasmid overexpresses a dominant negative version of p53, and it's quite effective at blocking the double-strand break response, and it's been commonly used. It was actually developed by the Yamanaka lab, and it's been used primarily to reprogram fibroblasts into hPSCs. So it's been used to make thousands of iPS lines. So it's kind of a standard reagent that we always use to make stem cells. So it's quite easy for us to test this in this assay. And here's the result. So these are pure-resistant colonies, and they're stained for a stem cell marker. If you look at the control cells, you can see there's a few colonies, but overall, most of the cells are not properly targeted and die once you add puromycin. When you look at the p53 dominant negative side, where we transiently inhibited p53, you see a dramatic boost in targeting efficiency. And this really says that the historic low engineering rates in stem cells are really because of double-strand break toxicity. How could you ever properly engineer when most of your cells die? And so p53 has a very strong effect in these cells. All right, to summarize. So p53 has been endearingly called the guardian of the genome. And p53 preserves genome integrity by triggering a toxic response to DNA damage, and this eliminates mutant cells. Our work in hPSCs highlights that mechanisms preserving genome integrity are directly opposed to our ability to engineer cells. And we demonstrated this by transiently inhibiting p53, which dramatically improved engineering outcomes in hPSCs, which is a cell type that has been historically difficult to engineer. We also think these findings have the potential to improve both the safety and efficiency of Cas9-based gene therapies. We think in the future it will be critical to ensure that patient cells have a functional p53 pathway before and after engineering. Okay, so for the second part of the talk, I'll go over genome-scale screening and hPSCs. And really the main question we want to answer here is, can we scale up, and can we have a high-performance screen? A really good way to do this is by running a fitness screen, and this is where we can use next-generation sequencing to look at the representation of guide RNAs in genes, and we can easily assess the dropout of core essential genes at the end of the screen. So there's really good annotated lists for genes that are essential in many cells. And also, while we're doing this, we also hope to identify stem cell specific genes, and this would mean that we have a platform that's really working and we can find interesting things related to stem cells. Another reason that we really focused on scaling this up is we had a lot of difficulties differentiating, editing and differentiated cells, and I'll talk about that in the final part of my talk today. And we thought, well, the editing is so robust in hPSCs and the mutations are permanent, so we wanted to really focus on refining this system and really get the performance up so we could have this bank of mutant cells that we could differentiate into neurons for our final screens. Okay, so here's the genome-wide screen that I conducted. I guess you saw earlier that toxicity was a big issue with engineering. But once you're aware of it, it's really easy to overcome. You just really need to screen more cells. So I think what's critical for stem cells or any cell that has a sensitive DNA damage response is screen a lot of cells. So we did this screen at 1000x. So for each guide RNA in the library, we had it 1,000 times. This is a big library. There's 110,000 guide RNAs in the library. There's 5 guides per gene. And we infected the cells at 0.5 MOI, so there's only 1 lentivirus per cell. So at the beginning of the screen, because we knew that our dox was not leaky and there was no CAS9 activity, we wanted to really bulk up the cells for a week before we started the screen. So we cultured them without dox for 1 week. This is large scale, so we had to grow the cells in these cell stacks. They're 5-layer cell culture vessels. When the stem cells are confluent, there's about 500,000 to 1 billion cells on each cell stack, and you put 1/2 liter of stem cell media into each cell stack. Okay, so at day 0, this is a really critical time point for the screen. We do 3 main things. One, we took a DNA pellet to look at the representation of the guide RNAs in our starting point for a screen, so we're going to normalize the remainder of the screen to that time point. 2, we plated new cells out at 1000x to undergo Cas9 mutagenesis. And 3, we did a pilot screen to see if we could actually freeze down our library and pull it out of the freezer without doing any bottlenecks. Okay, so we treated the cells for 12 days with Cas9, and this is when editing kind of is complete, actually a couple days passed. And then we took DNA pellets at day 8, 14, and 18, and we look at the representation over time using NGS. All right, so first we want to see, can we freeze and bank our libraries of cells, and that's shown here. So this is a log to full change for the 110,000 guide RNA library, and this is all normalized to the plasmid. This is really the standard way of doing it. And you can see our day 0 time point, there's a pretty big spread, but when we put cells in and out of the freezer at either 700 or 2100x, you can see that they maintain representation and things don't really change. So we have the same kind of spread. So this is nice, and it'll give us a lot of flexibility where we can do the infection one time and go back to the stable source of cells that we can start screens with. So really excited that we can actually do this and maintain representation. All right, now because we have this inducible system, we can actually do a better job of normalizing our screen to the no-dox samples, the actual cells you have in your screen instead of the plasmid. And so that's shown here. So when we normalize to our no-dox treated samples at day 0, you can see that these freeze-thaw samples get very tight. And when you add docs over time, you can see that there's an enrichment in dropout of guide RNA representation. So this is a nice way for us to normalize our screen, and it kind of cleans up the data because there's several issues between, chip synthesis that creates variability, the packaging creates variability, and the actual infection creates variability. So now we can kind of correct for all of that with this inducible system. So that's great. All right, so now we want to start getting into the gene level calls. And so, like I said earlier, there's 5 guides per gene in this library. And we can do 2 things to kind of measure their effect. We can use a quartile-based Z-score, and this will give you the magnitude of the effect for the 5 guide RNAs. We can also do a RSA test, which is redundant siRNA assay, which is developed for siRNAs because often with siRNAs there's a variability in the reagent, so you screen with like 10 or 20 siRNAs for a screen. And this allows you to calculate a p-value based on how tightly the guide RNAs cluster, and allows you to kind of throw out reagents that don't work. Okay. So how well is our screen working? So for this graph, we wanted to look at these gold standard gene sets. So each dot is a gene, and we only have 2 categories here. There's a set of non-essential genes that aren't expressed over 20 different cell lines, and then so they have no function really in most cells. And then the essential genes, which it's an annotated list of genes that drop out when you knock them out, collected by RNAi and CRISPR data. So we have the RSA down score on the y-axis and the magnitude on the x-axis. And you can see in the untreated samples, you can see that the non-essential and essential genes really cluster together. They're on top of each other, and most of them are above this cutoff at 10 to the minus 3 for the RSA score. Now, when you add docs for 18 days, you can really see that these 2 groups are separating. The essential genes are starting to drop out, whereas the non-essential genes have a nice tight distribution. So this is really kind of a qualitative look. Like, you can tell that you're getting what you'd expect. Essential genes are dropping out. We're having pretty good performance, but we'd like to do a more quantitative measure of how our screen is performing. So we employed this Bayesian analysis of gene essentiality algorithm. So this is really for the CRISPR screening aficionado, so I'm sorry. But basically this algorithm takes gold standard gene sets, and so we have these lists of essential and non-essential genes. And if you did a pooled screen, you know that the non-essentials have a very tight distribution and the cells are just growing normally, whereas the essential genes have a broad distribution and things are starting to drop out. So you have this training set in your screen, and you can use it to calculate this Bayes factor, which is the probability that your gene is essential over it being non-essential. So the higher the score, the more likely your gene is essential. Okay, and then you can generate a ranked list for all the genes in your screen. And you can use this to calculate precision and recall. So precision is the true positives over the true and false positives, and recall is the true positives over the true positives and the false negatives. And I'll just show you some sample data so you can see what this looks like. So we have 3 lists and 3 graphs associated with the list. So the essential genes are in red, and the non-essential genes are in blue. And what you can see is when they completely don't overlap, you have a very nice precision recall curve like this, and this is a perfect score. But as you have more false positives coming into your screen, like this non-essential gene here, your curve gets worse. And the worse your screen is, your curve looks more like this. It's not as nice. Okay, so how did our screen look? Oh wait, before we back up. So the group that developed this had this nice paper here measuring error rates for genomic perturbation screens, and they used this to analyze this very famous CRISPR screen before they even conducted their first CRISPR screen. And this is Feng Zhang's lab screen, and here's the precision recall curves generated by the Moffitt group. So you can see that the CRISPR screen done in the cancer lines, the A375, had a very good performance, and this is what you'd expect to see. Many essential genes are dropping out, but when they did it in stem cells, the performance was not so good. And there's 2 reasons for this. Maybe stem cells are very different from cancer genes, or the performance isn't good. We'd think it's the later latter because OCT4, the master regulator of pluripotency, didn't even drop out in this screen. So how does our screen look? So here's our untreated samples on the left, and you can see this is a terrible precision recall curve because it's just a random list. But when you add docs, you can get a very nice curve. And really the main difference between our screen and Feng Zhang's screen is that, we did our screen at 1000x and we avoided any bottlenecks associated with Cas9 toxicity, and that allowed us to really have a really functional platform. Okay, so I'll briefly go over stem cell specific hits. Now that we have this high performing gene, do we find stem cell specific hits? So on the dropout side, we have a lot of essential genes dropping out in pink, but you also see these gray genes. And these are putative stem cell specific genes. And importantly, we're able to actually find OCT4 dropping out, which is the master regulator of pluripotency. Now on the enrichment side, we get a lot of hits that aren't really annotated for any known function, but you can see that P53 dramatically enriches, and this is fitting given the toxicity story. So we think this is a rich data set, and we're going to find a lot of new and interesting things about stem cell biology as we get through this hit list. Okay, I'm going to briefly discuss some of the troubles we've had editing in postmitotic neurons here, and then I'm going to hand it over to Charles. Okay, so we've been having issues editing in differentiated neurons. So I have this pretty complicated system, but one of our favorite differentiation protocols is this DOX-inducible NGN2. And we just basically overexpress NGN2 in stem cells using the DOX system that I described earlier. And when you add DOX, you express NGN2 and you can quickly generate neurons. And then we wanted to multiplex this with our shield-inducible Cas9 so we could edit as stem cells or neurons as we wanted. So this is a pretty awesome differentiation protocol. In just 15 days of DOX treatment, you can have these beautiful neurons that stain for neuronal markers like MAP2 and CACNA1C. So as we were testing editing during differentiation, we saw that all these cells are highly transfectable. Pluripotent stem cells have a lot of RFP, and this is from the lentivirus. As you're differentiating, when you start adding DOX, you can infect, and also the more mature cells. But if you look at editing efficiency, you can see that this really drops off very quickly. So this lentivirus was added just 4 days after DOX induction, and Cas9 is not working at all. Okay, so what's going on here? Now I'm going to summarize a lot of different Cas9 transgenes, but we found that nuclear localization was very critical. So the first pair of Cas9 we were using was actually cytoplasmic, and it didn't edit in neurons. And then we further optimized this and got a Cas9 that actually could get into the nucleus here. And there's functional consequences to the localization of Cas9. So this second Cas9, that's more nuclear, edits much faster than the cytoplasmic one. This is indels over time. And if you look at the toxicity phenotype in stem cells, you can see that a targeting guide is toxic when it's cytoplasmic, but it's much more toxic when Cas9 is in the nucleus. So we kind of preferred this one unknowingly because it looked to be less toxic in stem cells, but then it didn't actually work in neurons. So, does this nuclear Cas9 work in neurons? So we tested it in a neuronal differentiation protocol. There's 4 phases of directed differentiation. At the end of 28 days here, we plate them in a 96-well plate. We infected with lenti CRISPRs, treated with DOX for 4 weeks, and isolated DNA. Now, when we first did this with the cytoplasmic Cas9, there was absolutely no editing activity, just like the NGN2 neurons, very disappointing. But when we used the nuclear Cas9, this dramatically boosted our efficiency, and we had an average editing efficiency of 66%. So this really made a big deal in postmitotic cells. In stem cells, they're cycling, so the nuclear membrane breaks down, Cas9 can get in, it doesn't matter. But when you're editing in differentiated cell types that become postmitotic, the localization of Cas9 is really critical to get the system to work. All right, to conclude, so we're getting a lot closer to realizing our goal of conducting genetic screens in human neurons. And our first screens will really be conducted by mutagenizing stem cells and differentiating into neurons. We have developed a really high performance system for stem cells, but unfortunately it's not one size fits all. It took a lot of tweaking to get this system to start to work in neurons, and we still have a little way to go. I suspect that, for other additional lineages, it'll really require cell type specific optimization of several things. For example, safe harbor loci, nuclear localization signals, guide RNA delivery, and a combination of all those things will enable robust editing. I hope that the points I've raised today can be used collectively by the hPSC community to develop a comprehensive set of Cas9 reagents that will really enable functional genomic studies in a wide variety of disease-relevant human cell types. Thank you. Lastly, I'd like to just mention that I'm very fortunate to be a postdoc at Novartis. I work with a small army of amazing people, and so many people have helped me out to perform these studies. It's very complicated, and it's a really collaborative environment. I have to thank my mentor, Ajamedi, for giving me the opportunity to be a part of his lab. And with that, I'll turn it over to Christy.
Thank you, Dr. Irie. As a reminder to webinar participants, if you do have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Mr. Joseph. Please go ahead.
Thank you so much, Christy, and thank you again for the opportunity to join today. So I just wanted to cover quickly a little bit about Twist Bioscience and the tools that we offer for CRISPR. For those of you unfamiliar with Twist, we are a synthetic DNA service company with a novel platform that allows us to write DNA on a synthetic surface. With the same footprint of a 96-well plate, our silicon surface contains thousands of clusters, each with 121 positions on which a unique oligo could be synthesized. And these oligos can be assembled either into genes or pooled for CRISPR library generation. Looking at the CRISPR library screening workflow, you typically design your guide RNA sequences, build them into a lentiviral library, and then test them to select for the desired function. And challenges can occur in maximizing screening efficiency and developing effective high-activity guides that maintain specificity. And Twist precision oligo pools can help address these issues. Assessing oligo representation in a pool of 29,000 oligos, we saw that on a global level, all oligos in the run were present and that 90% of them had an abundance that was within 4x of the mean, indicating a high synthesis uniformity. And you can see on the right side that the same observation was consistent on a per cluster level, not just globally, so there are no concerns of positional effects. Up for the same pool, we looked at error rate of synthesis, and using NGS, we saw that the error rate was 1 in 500 bases with just standard off-the-shelf phosphoramidite chemistry. And additionally, we looked at this at a per cluster level and saw similar results to uniformity. GC is often a concern of users on whether a high or low GC oligo will be successfully synthesized. So the same pool that we had designed previously included oligos with 15 to 85% GC, and we saw that there was minimal impact of the synthesis success based on GC content. A collaborator of ours, Kasuke Yusa from the Sanger Institute, compared the performance of a Twist oligo pool with that of an array-based competitor when building a whole genome CRISPR library, and he used about 5 guides per gene target. Looking at the experiment, the competitor pool not only had a wide distribution of reads, which was about 6 logs, but it also contained a long tail of low coverage reads for a portion of the library. We see that the Twist main pool maintains a much tighter distribution of reads across the entire library, about 4 logs, and with no low coverage tail, delivering a more efficient screen, and this will ultimately help save on sequencing costs as you only need to sequence to a lower depth of coverage to get all of the guides in your library. So Twist delivers a highly uniform oligo pools with very low error rates for precision CRISPR library generation. And if you have any more questions or interest in working with Twist and our oligo pools, feel free to contact us. And with that, I'll hand it back to Christy.
Thank you, Mr. Joseph. Before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. Okay, we'll now start the Q&A. The first question is for Robert. Do you see any toxicity when transducing with dead Cas9 effector proteins, for example, CRAB or VP64 fusions?
We've done less work with that, and I would say no. We're still trying to get that system up and running. Like, we've been making the cell lines, we've been expressing it and looking at the localization, and the short answer is no. We have done less with it because, you know, we have to, it's a whole set of new reagents, we need new guides, and it's just taken a little bit of time to get up. But from the little bit we've done, no. I mean, Bruce Conklin wrote a paper in cell stem cell with the CRISPR system, and it worked pretty well in pluripotent stem cells, so they didn't mention any toxicity. I doubt that there would be. I mean, for example, when you're overexpressing Cas9, you're inducing Cas9, you have a non-targeting guide that doesn't cut, there's not a lot of toxicity with that system. I'm not sure that the CRAB or VPR domains would really make that worse. Primarily the toxicity that I'm seeing is because of the double-strand break, and I'm sure most of that would go away with CRISPR-I. The reason that I've chosen not to pursue CRISPR-I is because we had a lot of difficulties engineering in neurons, and the CRISPR-I system would have those same problems. It could work really well in stem cells, but it would get turned off during differentiation. And the advantage with nuclease, it's a permanent mutation. So that's why we've stayed away from that, but I definitely would like to get a really fully functional CRISPR-I system, and we could avoid some of the toxicity issues, especially if we can get it to work across many cell types. Then I think it's the way to go.
Thank you. The next question is also for you. How do you monitor and possibly control collateral genome damage from the transient suppression of P53?
So, we're thinking about doing this. This will take, quite a bit of work to do. So, I think what one really needs to do is a tedious study of putting the pluripotent stem cells through clonal selection, plus or minus editing. Maybe pick hundreds of clones in either case and see and do deep genome sequencing and see how many mutations pop up. And you can't do this for all studies, but maybe, for one cell type that you're interested in, you can do it once and at least define some rates of off-target effects. And the other thing, that I think, certainly a possibility is for the selection for P53 mutations. We haven't observed this yet and we haven't done the experiments, but you would assume that, I've shown you that the mutants really suppress the double-strand break response, so there must be some pressure for those to arise from the population. So there's also a risk for that, which P53 inhibition could prevent. But we need to really balance those two things. It's basically the genomic burden versus the spontaneous selection of P53 mutations, and that's going to be a big study, and it's going to take a bit of time. One thing that could make things better is to, have improved ways of transiently inhibiting P53, such as, a small molecule, but not a lot of effort has been invested in that because everybody wants to activate P53.
Do you get any differentiation-dependent silencing of TET-on in the iCas9 system?
Yeah, definitely. That's probably why we're not getting 100% editing. So our system gets turned off, and that's well known with the DOX-inducible system. So basically, it's something that we need to get away from the DOX-inducible system to avoid that. It makes it harder to. That's why it's not working to maximally in neurons. So that's definitely going on.
And when do you foresee bringing this technology to clinical trials? Is toxicity contained to safe enough levels for in vivo therapy?
I think, you know, there's definitely room for this in the clinic. You just need to monitor this acute aspect of toxicity. I think there's a potential to select for p53 mutations, but you know, we really just need to sequence patient cells before and after. And I think there's certainly limits to the number of edits you can do. I know for T cells, it's been proposed to make CAR T cells with up to 5 edits. And I think that could be very dangerous. The more double-strand breaks, the greater pressure for p53 mutations. So I think if you have, you know, 1 or 2 sites you want to engineer and you do a lot of sequencing of p53 and actually the entire pathway before and after engineering, you can probably manage the risk and it'll be okay. But if you want to, you know, have T cells with 5 or 10 cuts, you know, I would really question how safe those cells are as they come and they survive that engineering process. I think that's quite stressful, and that's when p53 mutations or related genes will really pop out of the population.
Robert, does the post-mitotic nuclear localization of Cas9 version work in all the differentiation states that you've looked at?
I mean, we've just done directed differentiation and NGN2, and it seems to work. You know, we still have those transgene silencing issues going on in either cases, so that's hurting our efficiency. But it does, you know, that pair of NLSs seems to stay nuclear in both stem cells and neurons. Yeah.
Are you aware of any small molecule p53 inhibitor that can be added to the hPSC culture to reduce the toxicity?
You know, we tried to use the pifithrins. One was quite toxic. Mu is quite toxic, and alpha has solubility issues. So to really use it at the concentrations that other people have used it at, it's a ton of DMSO. Stem cells don't like DMSO, and you can see it precipitating when you put it in culture. So that's the problem with that. In our media, it drops out, and it didn't work for us. But you know, done a few studies on that. I mean, I kind of got away from that after seeing that it looked like it'd be challenging. I'm sure maybe if you optimize a lot, you could get it to work, because it does work in mouse cells. Where they were discovered and optimized, and it does work in blood cells. But for human pluripotent stem cells, we didn't have much success with that, but I'm sure it could be optimized.
Charles, the next question is for you. What's the mass of each oligo in the pool?
So for the pools that we delivered, you'll receive at least a quarter femtomole per oligo. And that sounds like quite a small amount, but we've routinely seen that it's sufficient for PCR amplification and cloning.
Thank you. Robert, back to you. HEC293 cells are often used to overexpress proteins. Would inhibiting TP53 during CRISPR editing improve bulk protein yields?
You know, so HEC293s, there's a couple versions. I know that one has the SV40T antigen, which kind of suppresses P53, and so it might already be kind of off in those cells. I know, and I think HEC293 doesn't have that. Maybe in the HEC293A cells that don't have P53 inhibition for their transforming, maybe it would improve yields there. But I think it's already kind of off, and engineering works really well in HEC293 cells, so it might not get any better.
And what media do you use to culture hPSCs?
We use E8, developed by the Thompson Lab, also MTeaser. We see similar effects in both of those two medias.
Charles, the next question is for you. What length oligos can you provide?
So our standard offering is 20 to 150 nucleotide oligos, and we are offering early access right now for oligos of length 151 to 200 mers.
And are there any sequence limitations on what I can order?
There are minimal sequence limitations because as I showed in the slide, that GC isn't really has much effect on the oligosynthesis. What has more of an effect is homopolymers. So if you start to get 5 or 6 of the same base in a row, that's where you start to encounter some issues. But other than that, it's really wide open.
Great, thank you. Robert, can you comment on the feasibility of modeling specific diseases with the help of CRISPR-Cas9? In particular, the person who's asking the question is specifically interested about Parkinson's disease.
Oh, certainly. I mean, I think the Yanish lab has already done that, and they've made, really they've made, SNPs and isogenic backgrounds using CRISPR-Cas9. So I definitely think it's feasible for Parkinson's. We had a paper, we did it, we used these same reagents to interrogate Zika receptor function, and that was in cell stem cells. So certainly it could be used for that.
Thank you. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. Charles, are there any options for ordering plasmid pool libraries or just oligo libraries that need to be cloned?
So we currently only offer unamplified oligos, but we have a partner in Desktop Genetics who focuses on guide RNA design for libraries. And through them and their partners, you would be able to get plasmid cloning services for an oligo pool generated by Twist.
Thank you. Robert, if you're stably overexpressing the CRISPR-Cas9 system in hPSCs, do you observe the same toxic response when you transiently express CRISPR-Cas9 using plasmids or RNPs? And could this be because of off-target effects?
Okay, so I have a few things on that. So we also made the same stable system with enhanced Cas9. We also saw toxicity. We sequenced off targets for some of the guides that we used a lot. We didn't see any off targets. The enhanced Cas9, if you don't know, is an improved version with a lot less off targets developed by Feng Zhang's lab. So that's in the stable system. Also in that HDR assay, that was all transient Cas9 expression in plasmids where we knocked in the transgene into the OCT4 locus. So that was transient, and we're seeing a dramatic effect when you inhibit P53. And then we spent a fair amount of time optimizing RMP delivery, and we'll get quite good editing with full-length guides. We're getting them from Synthego and Cas9 protein from IDT. We could just order those reagents, and we're getting around 80% editing for a couple guides. And if you look when the efficiency is really high, you can see cells die, P21 comes on, and that's really transient. I think in about 18 hours when you do RNP delivery, there's 50% editing, and it happens really fast. And that is supposed to really limit off targets, but we still see the same effect. So I think it's just a few double-strand breaks is really causing the stem cells to die in this case.
Do you know of other cell types that exhibit a strong toxic response to Cas9?
Yeah, so the cancer cells that we had access to data didn't have a strong response, but if you look at the 2014 paper by Tim Wang in the Sabatini lab, you can see they actually did a CRISPR screen to study the DNA damage response, and they used, I believe, HL60 and KBM7 cells. And they actually are very sensitive to DNA damage, and they die at the same concentrations of etoposide that stem cells do, so that's a DNA damaging agent. So they're very sensitive. And if you look at that screen very carefully, you can see that non-targeting guide RNAs really enrich in that setting too. It didn't get mentioned in the paper, but they were seeing the same thing. I think when you look at the methods for that paper, they also did their screen at 1000x, and they had good performance because I think they must have been aware of the same issue, but they just screened enough cells, and it really didn't affect the results, and they did a fabulous screen, but they definitely would have observed the same thing. I haven't tested those cells directly, but you can tell from reading the paper that it was going on.
Charles, the next question is for you. How many guides per gene do you recommend for CRISPR-I and CRISPR-A screens?
So we don't have any real firm recommendations. I think it just depends on the informatics that you're able to do downstream and how confident you are in the guides that you have designed. So if you know that the guides you've designed are very specific and they have minimal off-target, you know, the number would be fewer. But everything in silico and in vivo is different, so that's why it's good to have some redundancy. And for the customers that we've worked with, we've generally seen that about 5 guides per gene is quite typical and then gives enough redundancy in guide RNA design to calculate and measure the knockout effect effectively.
And Robert, can you comment on what G protein coupled receptors were critical for cell viability during the genome-wide screens, and were there any differences between hPSCs and cancer cells?
I cannot comment on that. I don't, you know, offhand I don't know, and of course any of the hits from that screen I'm still working on actively, so can't make any comments there.
Okay. Can you describe the key difference between your genome-wide hPSC screen and the one done by Feng Zhang's lab as described in an analysis by the Moffitt group?
Yeah, so I guess, you know, the key thing, I mean, we're doing this in 2017 and they did it in 2014. So they were really on the edge. And, you know, like I said earlier, it's all about being aware of toxicity and planning for it. So their screen was done at 270x, which is totally fine for cancer cells. And but it doesn't work for hPSCs because of toxicity. And you can understand why they did 270x, because the stem cell media is very expensive. So going to 1000x, you know, might have been cost prohibitive for them. So I think that is the major difference. And then there's a couple things with the type of the transgenic system. So we made a stable line, we picked a clone that expressed CAS9, knocked into the safe harbor locus. So that has very consistent expression and activity of CAS9. And then we delivered our guides with the lentiviruses, and we're getting, you know, 90% editing. I don't know what the editing for their system was, or if they even checked that. I think they might have just jumped into the genome-wide screen, but they're using CAS9 in guide RNA, you know, lentivirus. So each cell had a different, you know, independent integration of CAS9 and guide RNA. So that also creates some variability in the screen.
And Robert, can you elaborate on TRE silencing that you see during differentiation?
You know, I have I can't say too much about it. I know that it goes off, and there's plenty of published reports on it. When I started having problems with that, that's when I shifted to really doing pooled screens in stem cells, because I was like, well, I have amazing editing. This differentiated problem is going to be a tough nut to crack, and no one's really done a great job of expressing transgenes stably throughout differentiation. It's really hard. So I've kind of avoided that problem personally. Our group's working on it to solve it, but other than, you know, it's well known that it gets turned off, that's as far as I've gone. I just realized that was a hurdle that will take a serious investment to solve.
Thank you, and that's all the time we have for today. We'd like to thank Robert Irie and Charles Joseph and our sponsor, Twist Bioscience. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.